Welcome to ePartsala. Today I am going to discuss about the restriction endonuclease and isoschizomers and new isoschizomers. First of all, I would like to give you some brief about what is the endonuclease, what its relevance, what are the different types of restriction endonuclease, as well as what is the usage of this, and at the end I will give you the summary. So initially, first of all, we should know why do we need restriction endonuclease. Basically, restriction endonuclease is also called as molecular scissors or scalpels. What does it mean? It means that suppose if you want to manipulate the DNA, or suppose if you want to do something with the DNA in order to understand what is the exact nature of the stretch of that particular DNA, you can use the restriction endonuclease or you can use the restriction enzyme. As I said you, that it's also called the molecular scissors because how it works. Let's say suppose there is a DNA double helix. And in the DNA double helix, if you want to cut a specific stretch of that helix in order to clone or in order to know what is the efficacy, what is the expression pattern, and how this particular fragment is able to change the gene per se, then you can use this restriction endonuclease. How it works? Suppose, let's say there is a DNA sequence, and in the DNA sequence, there has to be some recognition site, and that recognition site can be read by restriction endonuclease, and it can recognize a particular base pair and then it cut. Second thing, there are different kinds of restriction endonuclease which has a different, it's called cutter. Like if the 4 base pair is called 4 cutter, 6 base pair is called 6 cutter, and 8 base pair is called 8 cutter restriction endonuclease. So let's start with some history and background of the restriction endonuclease. Restriction enzymes were first identified and discovered by Werner Arber in early 60s, and however it was not that means like it clicked. So what happened that in 1970 Nathan and Smith first isolated the restriction endonuclease successfully, and for that in 1978 Nobel Prize has been given to this trio, that is Werner, Nathan and Smith. Nomenclature part, let's say suppose if you heard the name of EcoR1. So how this EcoR1 works? Let's say EcoR1, E stands for Escherichia, CO stands for coli, which is derived from the bacterium, so E. coli, and R is a strain, and 1 is the first enzyme isolated from that particular species, and that is the why it's called EcoR1. Likewise, Hint3. Hint3 is coming from the Haemophilus influenza, and the D is the strain type, and third means that it comes from the same bacteria, and the nomenclature is third because it's the third isolated restriction enzyme. So I can give you the glimpse of different restriction enzymes like EcoR1, Hint3, BAMH in a consolidated table, that okay, what is the genus, what is the species, strain, what is the number of isolation, and you can also see the recognition sequences of these restriction endonucleases. Now come to the point, what is the type of restriction endonuclease? And here you can see there are different types of the restriction endonucleases, right? Type 1, type 2, type 3 and type 4. In type 1, it's a cleave at random site, means it can be 1000 base pair away, or it can be upstream or downstream. So this restriction enzyme type 1 is not that very specific, and it has the different characteristics such as it has multiple subunit complex, and there are different cofactors which is called S-adenine methionine, different salt ions, and as well as ATP. Now come to the type 2, and type 2 is the most important restriction endonuclease which is useful for the DNA manipulation. Let's say it's a very specific, first slide on the DNA stretch, recognize their sequence and cut in palindromic way. What is the palindrome? Palindrome means that sequence should be 5' to 3' is exactly like in the antisense strand. Suppose if GAATTC is the 5' to 3', if you read in the antisense strand from 5' to 3', again it will say like GAATTC. I'll explain you this later, but here this is the most specific restriction endonuclease which is useful for the DNA manipulation, cloning and many more purposes. Come to the third category, that is a type 3 restriction endonuclease, and here you can see it cleaves outside in the recognition sequence and require 2 restriction sites in opposite directions. Okay, and the characteristics of this restriction endonuclease type 3 is it can modify the enzyme, cut at different location within the 25 base pair region. Fourth type of the restriction endonuclease is type 4, which is basically cleave the methylated DNA, and it also requires magnesium ion as a cofactor. Now come to the overhangs. First of all, you should understand what is the nature of restriction enzyme first, second thing how it cuts, and third, if it cuts, what kind of overhang it produces. So basically there are three different types of restriction enzyme. One is a sticky end, it provides a sticky end. Second is the staggered or blunt end, and third is the 3' overhang, it's also a sticky end. So if you can go with the 5' overhang, and denotes all the four different nucleotides, that is A, T, C and G. So 5' overhangs recognized by the restriction enzyme in the 5' area, and it cleaves in such a way that it can give a 5' overhang. Likewise, in the 3' overhang, it can cut from the 3' direction and it give you the 3' overhangs, while if it's in between the DNA sequence, it can give you the blunt end. So now you understand what is the 5' overhang, 3' overhang and blunt end. So here I can give you the consolidated list of some important restriction endonuclease and their source, recognition pattern, that is 5' to 3', and what is the base pair and overhangs. So if you can go one by one, PVU, it gives you a sticky end as a 6-cutter restriction enzyme, while as EcoR1, BGL1, HIND3, BAMH1, SAL1, these all are the 6-cutter restriction enzyme and give you a sticky end with the different overhangs. Now come to the 4-cutter restriction enzymes, and 4-cutter restriction enzymes are SAL1, TAC1, HAE3, these and HAPA1. These are 4 base pair long restriction endonuclease type 2. You can see that in the 4-cutter restriction enzyme, you will have again the blunt as well as the sticky end. Now come to the 8-cutter restriction enzyme, which is the NOT1. And you can see that this is the 8 base pair, and this 8 base pair can cut in a sticky end fashion at the 5' overhangs. So this list can give you a detailed idea what is the overhangs, what is the ends, that is whether a sticky end or a staggered end, and different recognition pattern from 5' direction to the 3' direction. Now come to the mechanism of action, how these restriction enzymes are working. And here you can see what happens. Suppose if you have a DNA molecule that has to be cleaved, and for that you add the restriction enzyme in a particular fashion. And what is happening here, these restriction enzymes first recognize their recognition site, which is present in the DNA stretch. Then what is happened, it binds to that DNA at recognition sequence. Then it gives a single cut to the sugar phosphate backbone in the double helix, means sense strand, as well as in the antisense strand. And fourth, it hydrolyzing the phosphodiester bond, and thus you can have a particular desired DNA stretch which you want to be cleaved. What are the different factors that accurately work, that enable restriction enzymes to accurately work? And for that you need the first and most important is the temperature. Some restriction enzyme can work at lower temperature while some in a higher temperature. So let's say the most common temperature for the restriction endonuclease is 37 degrees centigrade. So you need a correct temperature to work for restriction endonuclease. Then salt concentration. Salt concentration is also an important factor for the restriction endonuclease. And third is the pH, because you have to maintain a physiological or balanced pH in order to cut the DNA very specifically and in a proper way. Fourth is the cellular homeostasis, because these restriction enzymes are derived from the microorganisms. So you have to give a condition which can mimic the biological or cellular homeostasis, so that it can say that okay, this enzyme is coming from one particular organism, and that is the reason why we have to give a particular homeostasis to have the best efficacy of the restriction endonuclease. Now we should understand, we have already learned 4-cutter restriction enzyme, 6-cutter restriction enzyme and 8-cutter restriction enzyme. Now how it works, what are the different frequency of these restriction endonuclease? Let's say 4-cutter restriction enzyme, which means n to the power 4, and here again, as I said in the previous slide, n is ATCG. So 4 to the power 4 is 256, which means any restriction enzyme which has 4-cutter can invariably or indiscriminately digest the DNA or chop the DNA at every 256 base pair, while in the 6-cutter again come to the power n to the power 6. So n to the power 6 means 4 to the power 6 is equals to 4096 base pair, and again here 4 kata restriction enzyme 256 base pair, 6 kata restriction enzyme 4096 base pair, and likewise 8 kata restriction enzyme will cleave 65,536 base pair restriction enzyme in a genomic DNA or in any big chunk of DNA. So now you understand how these restriction enzyme has the frequency to cut or chop the DNA invariably, indiscriminately. Now we should understand what are the different usages of the restriction enzymes. First of all, as I explained you in a previous slide, restriction enzyme is used to manipulate the DNA for cloning purpose. Let's say we want to check expression artificially. We cannot do every time inside the body because it's come under gene therapy, and gene therapy is not that easy. Even in the gene therapy, we have to manipulate the DNA, and for that you need restriction endonuclease. So what happened here, that first of all we have to manipulate the DNA in order to prepare or in order to generate clone. Second, checking the polymorphism. How you check the polymorphism of a gene, and this can be easily done by restriction endonuclease. Let's say example of the RFLP, restriction fragment length polymorphism. Third is the forensic science. Like you have heard about the DNA fingerprinting. DNA fingerprinting is exclusively be done by the different restriction enzyme. Third is the restriction mapping. Suppose if you want to know what is the exact physical map of a particular DNA stretch, you need restriction enzyme which can give you a particular physical, not particular, exact physical distance of DNA. Third is generation of the DNA libraries. Generation of the DNA libraries is also important in order to understand the gene expression or gene profiling, and for that you again need the restriction endonuclease, and this restriction endonuclease can help you to prepare different kind of library, whether it's a cDNA library or it's a DNA or a genomic library. Now come to the last point, that is the creation of the gene chip. Even you can heard about a lot of gene chips which can give you information at one time and huge number of information, and again here you need restriction endonuclease in order to understand better how these gene chips are working. I'll explain you one by one. First start with the DNA manipulation and the cloning. So you can see that suppose if we have prepared an artificial vector, for that again you need a restriction endonuclease, and suppose if you have a artificial vector, subtle vector or any kind of vector from any origin, it has some promoter region, poly A region, origin of replication region and antibiotic resistant region, and here you can see that you will have a multiple cloning site which is called the MCS or polylinker, and this polylinker is having the array of different restriction enzyme which is compatible in such a way that you can clone in particular fashion. Let's say suppose you can see here there is 10 different examples where you can see different restriction enzyme in per fragment, and every fragment you have a different set of restriction enzyme. You have to match this restriction enzyme strictly in a 5' to 3' orientation. If you want to prepare an antisense cloning, you can go by the 3' to 5', but if you prepare the sense clone or physical clone, then you have to go from the 5' to 3' direction, and you have to understand why these different fragments are able to give you the exact nature of gene expression. So these are the 10 different examples, and you have to match from the 5' to 3' direction in the vector. So lower panel is the insert, while upper panel is the subtle vector with the multiple sequence of multiple cloning sites, and here you have to add these two, that is called the ligation, and then you can generate a foreign particle in a particular vector, and this is called the clone or construct. Second is the RFLP. Suppose if you have a isomer, or if you have isoforms of a very closely related gene, let's say talk about the cytochrome P450. Cytochrome P450 superfamily has a 2C subfamily which has different genes, and these genes having the peculiarity of 90 to 95% sequence similarity. Suppose if you want to pinpoint exactly what is the real difference between one isoform CYP2C isoform with a different CYP2C isoform, you can use the different restriction endonuclease in order to understand the RFLPs. And here you can see that when you can see in both the slide, when we have digested the genomic DNA using restriction endonuclease, 4 kata restriction endonuclease, we have got this pattern, and after sudden hybridization, the in down panel, you can see that you have got some different signals, which is different than the all other signals. And I have given you a small stretch of homologs, that suppose there is a 6 kb homologs of DNA stretch, and the 6 kb homologs, if you have different, if you have a liberty to use different restriction enzyme, you can chop that DNA very easily, and you can say that, okay, what is the real probe we can generate in order to isolate or fish out the particular or gene of interest in a big chunk of DNA. So this can be used for the restriction fragment length polymorphism or polymorphism per se. So the third usage of the restriction endonuclease is restriction mapping. So suppose if we have a very big chunk of DNA, let's say 30 kilo base pair DNA, and in the 30 kilo base pair DNA, suppose if I have only one restriction enzyme, we can go for cleavage, and in the cleavage it can give a 2 or 3 different fragments. Like in the you can see in the right hand panel, the enzyme 1 has given 6, 3 and 8 kilo base pair fragments, while enzyme 2, I mean restriction enzyme 2, is given 7 and 10 kilo base pair fragment. But what is in between? And you can see, suppose if I can use the enzyme 1 and enzyme 2, what would happen? You can have intermediate sequence which were masked in single digestion, that is the using of this one restriction enzyme. So instead of using one restriction enzyme, if we can go for the 2 restriction enzymes, we can have the better understanding of the physical map or physical length of a particular DNA. Now come to the point, isoschizomers. What is the isoschizomers? As you can see, iso means similar, restriction enzyme that have the same recognition sequence as well as the same cleavage site. And when you have this kind of architecture of a DNA, or if you can have these kind of possibility, this is called the isoschizomers. Example for the isoschizomers is SPH1 and BBU1, and here you can see the recognition sequence is GCCG, TACG in both the restriction enzymes, and the recognition pattern is 3' overhang at G. You can see it here very clearly that although these are the 2 different restriction enzymes, but the recognition sequence and cleavage pattern is same. Definition of the neo-isoschizomers is the restriction enzyme that have the same recognition sequence but cleave the DNA at a different site within that sequence are called neo-isoschizomers. And the best example is SAMA1 and EXMA1. I want to explain this EXMA1 and SAMA1. EXMA1 is a 6 cutter restriction enzyme, while SAMA1 is also a 6 cutter restriction enzyme, but the EXMA1 cut in the 5' overhang fashion, that is the sticky end, but the same sequence when it cut by the SAMA1, it will give you blunt end. So EXMA1 and SAMA1 is the best example of the neo-isoschizomers, where you can see that at one end it gives you 5' overhang, while on the other hand it can give you the blunt end, means a sticky end as well as the blunt end. The third type is isocaudomers. It's a pair of restriction enzymes that have slightly different recognition sequence, but after cut generate identical stretch. What does it mean? Like suppose there are 2 6 cutter restriction enzymes called MBO1 and BAMH1. So MBO1 and BAMH1 in the initial nucleotide, which is denoted here is the N, is different with the BAMH1, but the cleavage pattern, which is also a 5' end and it's a sticky end, and it gives GATC, which is similar in the both the sequence, that is MBO1 and BAMH1. So what is the difference? Difference is only the initial nucleotide, which can be different, but the cleavage pattern is similar in both the restriction enzymes. So before concluding my talk, I would like to give you the glimpse of restriction endonuclease as well as the isoschizomers, neo-isoschizomers and claudomers. So first, restriction enzymes are digesting DNA only at a specific recognition site. This is the most important thing, and therefore it is quite manageable to understand the physical length of a DNA fragment within the boundary of 2 restriction sites. What does it mean? It means that this is not only give us the precise information and sequence of DNA, rather enable us to manipulate the DNA with desired gene of interest. The second point, that classification, and classification is like that. Restriction enzymes, as I said in the beginning, that broadly classified under 4 categories based on the proximity of recognition site and cleavage pattern. Moreover, the restriction enzymes digests DNA double strand by making 2 incisions in sugar phosphate backbone on each DNA strand. Third point is, what is the difference between restriction enzymes and modification enzymes? So for that restriction enzyme cuts the DNA strand within the sequence. However, there are different DNA fragment which has to modify in order to cloning purpose, and for that the enzymes which modifies the various ends or the 2 different ends of DNA is called modification enzyme. For example, T4 polynucleotide kinase, which adds phosphate at 5' end, whereas alkaline phosphatase, which is removing phosphate from 5' ends. Likewise, ligase, it can join 2 DNA fragments during the cloning process, as well as this T4 DNA polymerase, which can fill in with the help of DNTPs. Another important point in restriction enzyme chapter is type 2 restriction enzymes are mostly palindromic in nature, which means the reading of sequence from 5' to 3' and 3' to 5' is same. For example, EcoR1, which is GAATTC. If you read this restriction site from both the ends, it will appear the same, and that is why it's called palindromic sequence. However, restriction enzymes produces 5' and 3' overhangs that I have already explained in my previous slides, that what has happened in the 6-cutter restriction enzymes when its incision is at the 5' end and at the 3' end, and thus it gives 5' and 3' overhangs, which is also generating, or which is also called the cohesive or sticky ends. However, sometime when the digestion is in between the equal number of nucleotide sequence, it produced the blunt or staggered end, and thus this is useful for the blunt end cloning. Now come to the next point, that the origination and source of the different restriction enzymes, and you can see in the table that I have already explained before, that what are the different bacterial source of organism through which enzymes have been produced. Enzymes have been produced. Second, the recognition sequences. Third, the different ends. What are the different characteristics of restriction enzyme, whether it's a 6-cutter, 4-cutter or 8-cutter restriction enzyme, and at the same time what kind of end it's producing, whether it's a 3' overhangs, 5' overhangs or blunt end. In the end, I just want to give that what this special restriction enzymes are doing, that this special restriction enzymes, where restriction enzymes that have the same recognition sequence as well as the same cleavage sites are called isoschizomers. For example, SPH1 and BBU1, where restriction enzymes that have the same recognition sequence but cleave the DNA at different site within that sequence are called neoschizomers. And another type where the pair of restriction enzymes that have slightly different recognition sequence but after cut generates identical stretch is called isochordamers. To summarize my talk, I can give you now you can understand the glimpses of the different restriction endonuclease. First of all, it digests the DNA at your desired recognition site, but only you have to know what is the exact DNA sequence. So suppose if you have the DNA sequence information, you can use a particular restriction enzyme which can cut in a very specific fashion of that DNA stretch, and it can give you the desired fragment, either for the cloning purpose or for the preparation of probe or for the any different aspect. Now come to the cloning and polymorphism. As I explained you, that in order, if you want to clone a foreign fragment of DNA in a particular vector, how would you do? Now you can do, suppose if you can digest a particular DNA stretch using 2 restriction enzymes, and if the same restriction enzyme is also present in the vector polylinker region, you can digest vector with the same two different restriction enzyme, you digest the insert with the same two restriction enzyme, you can clone it, which is called the ligation. Third thing is, let's say you have a PCR amplicon, and if you don't have any restriction enzyme for your convenience, what you can do, you prepare a primer in such a way that you can load the restriction enzyme on the forward primer as well as on the reverse primer, and what you can do, you can just amplify the product, and the amplicon you can digest with that particular restriction enzyme which you have loaded already, and the same restriction enzyme, if present in the vector multiple cloning sequence site, you can easily clone the amplicon which has already been digested with the two restriction enzyme into a vector. But before any cloning, you have to prepare a cloning strategy, and that cloning strategy will tell you what two suitable restriction enzyme, because for the cloning you must have two identical restriction enzyme present in the vector as well as in the insert. But the most important thing, that particular restriction enzyme has the unique site. What does it mean, unique site? Unique site means that in the entire region there has to be only one restriction enzyme in the insert as well as this one time restriction enzyme in the vector. Suppose if you have the two different one restriction enzyme with the two different sites, it will cut more fragments. So in order to clone, you must have two identical restriction sites in the insert as well as in the vector, which you have to prepare in advance while you are doing the cloning strategy. Come to the DNA sequencing. For the DNA sequencing also, you need restriction enzyme in order to cut. When the genome sequencing has been accomplished, it was done like that. So you must have restriction enzyme in order to make the very humongous DNA into a short stretch. And these short stretches will give you more or better idea how to learn the DNA stretch, and it can be achieved by restriction enzyme. Come to the physical map. Suppose if you don't know what is the exact physical map of a DNA, you can use the restriction enzyme. Suppose if you can take one restriction enzyme, let's say one particular restriction enzyme will cut DNA 3 times. Second restriction enzyme will give you cut 4 times. So suppose if you will 3 fragments or 4 fragments, you would like to know what is the exact physical map of a particular DNA stretch in between these two, or overlapped sequence or masked sequence. And in order to understand the masked sequence, you must have this kind of restriction enzyme. It will give you the correct physical map between the two restriction enzymes in a particular fashion, and it will give you many other informations. Now come to the construction of genomic library or cDNA library. Again, for the cDNA and genomic library, of course, for the cDNA library you have to go for the RT-PCR, and you can have the single strand DNA, and from there you can go for the cloning purpose. But again, if you will have the convenience of the restriction enzyme, the clone can be much easier, as I explained you in the case of amplicon, where you can load two different restriction enzyme in the forward region as well as in the reverse region. That is, what does it mean? Forward primer as well as the reverse primer. So you have to load the different restriction enzymes which is compatible with vector on the forward primer as well as the reverse primer, and this approach can also be used in the cDNA library, where you should have a particular length of a DNA that you can easily or conveniently cut with a restriction enzyme. So you may or may not use. Now come to the generation of artificial vector. Suppose if you want to prepare an artificial vector, that vector has to be carried lot of different restriction enzyme in the polylinker region, and for that you would again need a specialized methodology of restriction endonuclease that can easily be accommodate these restriction enzyme in a particular vector. And again, like for the chip, suppose if you want to prepare a chip which has a lot of DNA molecule, if you can digest this chip with the restriction endonuclease, it can also give you information that how it works properly in a small fragment. If you want to prepare a probe, you can prepare a probe by the digestion of the different restriction endonuclease. Thanks.